Transcriptional profile analysis of peroxisome-depleted Drosophila melanogastor macrophages. Another day, another COVID-19 news article to read with my morning coffee. The pandemic's biggest mystery? Let me guess, the virus? Nope. To my surprise, Ed Young from The Atlantic instead discussed the world's biggest mystery as our own immune system. Despite the field of immunology being traced as early as 430 BCE, the 2020 pandemic has only reminded us of how little we may know about our own immune system. This is where a small organelle comes into play, the infamous peroxisome. This organelle plays an important role in peroxide metabolism, fat digestion, and detoxification. However, little is actually known about this organelle's full capacity in the cell and the immune system. The Cara lab has identified that the peroxisome is essential for the phagocytosis of microbes to occur in macrophages. However, the specific mechanisms are still under question, which is where my work comes into play. To prepare for my analysis, the Cara group probed control macrophage samples and peroxisome-depleted samples in both control and bacterially infected conditions. This technique allows us to tell which genes are turned on or off in the cells. I use differential expression analysis on the sequence data to apply statistical models and quantify significant changes in gene expression. Once the preliminary analysis is complete, as shown here by the DESeq2 generated map and table, the data is then visually analyzed using pathway analysis. Bioinformatics software like Cytoscape generate visual maps of the genes enriched and connect them all in pathways to help us understand major biological themes occurring in these cells. By comparing the genes upregulated in the control sample and downregulated in the peroxisome-depleted sample, it is evident that these genes are involved in the Rho and Ras GTPase signaling pathway and in the Toll and IMD pathways, amongst other cellular functions, of course. These pathways are all pivotal for the macrophage's response to foreign antigens. Thus, one of my conclusions from these maps is that the absence of a peroxisome in a macrophage not only affects phagocytosis, but also in known immune signaling pathways. This summer has really opened my eyes to the realm of possibility in the research field. I learned so much from Dr. Cara and so much about myself along the way. I learned how to code, write lab protocols, practice my presentation skills, analyze data, and I even learned how to master in-lab techniques that were used to create my profiles. Hopefully, the next time you take your morning coffee and sit to read the dreadful pandemic news of the day, you can think of this tiny organelle in your cells that may be one of the keys to solving the mystery we know as our immune system. Thank you for listening.